Hello, everyone. Welcome to Antibodies, Next Generation Proteins, and Bioconjugates Digital Week, brought to you by the producers of the face-to-face Antibody Engineering and Therapeutics events. My name is Michael Keenan. I'll be your host for today's session. First, I'll cover some quick housekeeping items. The first thing, if you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, you can submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of this session. You can also download a featured white paper in the resource list box on the right side of your screen. Let's now begin by introducing our speaker from Twist Bioscience, Aaron K. Sato, PhD, Chief Scientific Officer, Biopharma. Thank you for joining us today, Aaron. Now I'll hand it over to you to begin the presentation.
Thank you, Michael. Again, my name is Aaron Sato. I'm the CSO of Twist, and I'm also the head of the Twist Biopharma vertical as well. So today, I'm actually going to give you an overview of our library of libraries. It's actually expanded quite a lot in this last year, and I'll actually roll out some new libraries today with some really awesome data associated with them. If I do have time, I will get into our optimization platform, but because our library is getting so big, I'm going to really focus in on that particular part of our discovery platform. So I always say the best companies out there really understand the one thing they're really good at. So Twist, that's our ability to print or synthesize DNA. And we do that on this silicon chip shown in the middle of this slide. Basically what the technology allows us to do is actually we can print up to 1 million individual oligos of discrete sequences in these tiny little spots on this silicon array. And this allows, it's basically a fantastic technology to make large pools of individual oligos. And we can use those pools of individual oligos to make all kinds of custom DNA products. And in particular, for the Twist Biopharma vertical, we lean on the parent company, the main company, Twist Bioscience, to help us make all these fantastic libraries I'm going to talk about today using this amazing oligo pool technology. So how do we do that? So again, we use these oligo pools, and we basically combine them together in different ways using constant synthetic DNA as well to give us these very precise libraries that have base-by-base precision. We can exactly choose all the codons we put into individual parts of the libraries that we create. In terms of making antibody libraries, as you'll see later, it's very easy for us to vary the length of oligos, like the CDRs in our antibody libraries. And we can, again, just do that by varying the length of the individual oligos in that particular oligo pool in that library. Since we're printing every single DNA base by base as we build the libraries, we can avoid restriction sites and unwanted motifs, of course, and we have precise control over the combinatorial diversity. We also build libraries that are often called ratio libraries, and here, within, for example, a defined CDR, we can control the ratios of amino acids very easily by printing individual oligos and pools that match that ratio that we want. We, of course, can use multiple germline scaffolds if we're making an antibody library. And then finally, at the end of the library process, we validate our libraries using next-generation sequencing. How do we use oligo pools to build antibody libraries in particular? So shown here is an example of an antibody domain. Let's say it's a VHH heavy chain only library. We can basically make oligo pools that constitute the diversity in each of the different CDR loops, and then to make the final library, we can piece them together through PCR. The cool thing about the way Twist builds libraries, in particular antibody libraries, is that when I make these oligo pools that constitute these diversities in CDRs 1, 2, and 3, I can synthesize sequences derived directly from natural human diversity. And so this allows me to shuffle CDR diversity that's naturally derived, and as a result, the antibodies that come out of these libraries look almost indistinguishable from anything that would come out of a human. So unlike a lot of our competitors that might use, apply mutations in a ratio-based library, they may actually, even though they might get really high affinity antibodies, they may also get antibodies that have unnatural pairing of specific sequences in their leads, which may be not derived from natural human sequence. I also can remove sequences up front that might lead to liabilities in antibodies that come out of my library. So I can basically remove CDRs, even though they might be naturally occurring, that have isomerization, cleavage, deamination, glycosylation sites. And then finally, as you'll see later, the cool thing about using alga pools to revealing antibody libraries is I also can insert specific motifs if I choose to. That's actually key and central to our GPCR library that I'll talk about later. So again, Twist Biopharma, we're a division of Twist Bioscience. We're focused on using our library platform as well as our ability to make clonal genes using the silicon array platform I talked about before to help others discover antibodies as well as to optimize antibodies. And as I said before, I'm going to really home in and focus almost exclusively on our antibody discovery product. If I have time, I will talk about our antibody optimization platform called Tau. But in the interest of time, I'm going to really focus in on our discovery platform. So at a high level, what do we really focus on within Twist Biopharma? First of all, I'll, as I said, really focus in on our library of libraries, which is a wide array of different libraries from Fab to single chain to VHH, heavy chain only antibodies, all in phage display where we can then pan and screen them against any target that we wish with our partners to help them discover novel antibodies to their targets. We're also working on a number of other approaches, which I won't talk about today, that are enabled by modeling or immunization to give us large repertoires. We can also make libraries against specific targets. As I mentioned and hinted before, the ability to use alga pools to build libraries allows us to make target-specific libraries. In particular, we've made a number of libraries that focus in on difficult to drug target classes, for example, GPCRs, which I'll focus on quite a bit today, ion channels, as well as carbohydrates. As I mentioned, we do have an optimization platform that, again, rests on top of our library platform called Tau, which, if I have time, I'll talk about. And then finally, all of the projects that we do rely heavily on the ability to make full-length IgGs. And so actually this year, we're rolling out a new alpha product called High-Throughput IgG antibody production. And so I'll briefly talk about that. And if you're interested in our High-Throughput IgG offering, please follow up with me. And again, that's enabled by our High-Throughput gene synthesis platform, which uses the alga pool technology as well as a huge amount of automation in e-commerce. So why Twist Biopharma? You're probably thinking, you know, there's a lot of antibody discovery companies out there. What differentiates you from all of your competitors? Well, the first one is, unlike a lot of our competitors, we are a DNA company. We have this amazing ability to print DNA using the alga platform, and that allows us to build, you know, tons of libraries. And that's, again, why we've created this, coined the term, the library of libraries. A lot of our competitors may only have one or a couple libraries that they use repeatedly over and over against different targets. But here, as you'll see today, I now have over a dozen different libraries that I can use in pools to really ensure that when our partners give us a target, we can give them antibodies not just from one library, but from multiple libraries. So that increases our broad epitope diversity of our antibodies that come out of our specific projects. Second, as I mentioned, when we create these alga pools to build these libraries, we can ensure that those sequences are derived from human sequences. And so that even though our libraries are synthetic, we can make sure that all the leads that come out of our libraries are human-derived and look almost exactly like you would see coming out of a human being. And then finally, Twist has this amazing ability to miniaturize and automate the entire process of antibody discovery and optimization, and that really rests on our ability to make DNA. And this really speeds the efficiency and overall speed of the process to go from initial target all the way to functional antibodies. So as I mentioned, I'm going to really focus in today on our library of libraries, which again is a wide array of different libraries using different diversities, some of which are generally used naive libraries, others are actually target class specific. So here's an overview of all the libraries that I've talked about before. And then as I said, at the end, I'll actually roll out some new data on 3 libraries that I've hinted at before in my previous presentations, but today I'll actually have some real data associated with them. The first of our libraries I've talked about before is a Hypermutated library. It's a fully human Fab library, again, in phage display, where we have diversity in both heavy chain and light chain. which is our original library, as well as a common light chain version of that same library for use in common light chain bispecifics. We also have a whole suite of different VHH single domain libraries. They're the ratio shuffle, human shuffle, Hypermune. We're also going to be rolling out a GPCR version of our VHH library later on in the year. I put it here just as a reminder. I won't have any data on that library today, but suffice to say we'll have a GPCR equivalent library for our VHH libraries coming out very soon. We do have a antibody library that we call our structural antibody library. It's an scFv library, again in phage display, where we basically take in a huge amount of diversity from all the antibodies in the PDB, and we use that as a design set to make a library with the idea that antibodies that have been crystallized and the structures determined are usually very stable and very developable. We wanted to focus in and make a library based on that diversity. And then the three libraries on the bottom are basically focused on difficult to drug targets. For GPCRs, we actually have two approaches that we've developed for libraries to target this difficult but very important class of targets. And then we also have libraries focused on ion channels and carbohydrates as well. So how do we go from initial target to discovered antibody? Well, one of the great things about COVID is that, there are not many great things, but one good thing about it is it actually pushed us to really speed up our overall process of discovery. And so we've actually been able to condense it down to an 8-week interval due to a lot of the work that we did against the COVID pandemic, which you'll see later some of the examples that I'll show for our libraries. And that really enables us to very quickly go from target to functional antibody in a very short period of time. Our process is we take your target, either cells or proteins, the protein target, we do panning and screening for 2 weeks to give us our initial hits. If it's a recombinant protein, we'll do an ELISA with thousands of clones to find unique hits. If it's a cell-based target, we oftentimes do NGS sequencing on the repertoire to see if there's enriched sequences that we might want to synthesize. After we synthesize those unique clones or do NGS on those outputs, we come up with a whole battery of sequences to reformat to IgG. We scale up those DNAs. Again, because we sit inside twists, we have this amazing ability to make clonal genes, and so we can do that very rapidly and with large numbers. So oftentimes we reformat up to 100 plus antibodies from every campaign, so that gives us a huge amount of diversity to look at. As I mentioned, we have this now amazing ability to make IgGs in high throughput as well, not just DNA. And so within a week, we can make antibodies derived from those sequences and then put them through a whole series of binding and functional assays. So in terms of that new high throughput IgG alpha product, so this year for select customers, we're offering up the ability to not only synthesize the DNAs that encode for specific antibodies, but also to actually make the antibody itself. So the idea here is that you can make either full-length IgGs of different isotypes that we already have set vectors for, as well as VHH FCs. We will take those DNAs that we, of course, synthesize in our silicon platform that I talked about before. We'll scale up the DNAs. We'll do transient transfections at HEC293. And then do that at either one of two scales. We can do a 1 mil scale, which typically gives us around 100 to 200 micrograms of either IgG or VHH FC, or an 8 mil scale where we can actually get upwards of even 1 milligram of antibody. And then we purify those antibodies on Pro-A tips to give us final purified antibody through a one-step purification. As I mentioned, we can typically get about 100 micrograms to a mg. An antibody can use two sizes and deliver that within around 20 business days. And we have two different buffers that we can use, one that's amine-based and one that doesn't have any amines for if you want to do any sort of downstream conjugation. So please follow up with us if you're interested in our high-throughput IgG offering. But as I said, it's built into our discovery projects as well. So now I'll jump into the different libraries in the library. This is a library that was actually our very first member of our libraries, and I've talked about it quite a bit in the past. I'll briefly talk about it now. So this is our GPCR 2.0 library. It's again a library focused on finding antibodies to G protein coupled receptors, and it's a motif-based library where it's based on this idea back in 2014 from Liu et al. And what Liu et al. Did is they They took a known crystal structure of CXCR4 bound to a loop peptide called CVX15. And when they hypothesized is, could they actually take this loop peptide, which is a known antagonist of the receptor, and actually graft it into an antibody, heavy chain CDR3, in the hopes that that grafted antibody would retain the binding and antagonist ability of that original peptide. And that's exactly what they were able to show. They were able to show that they could take that peptide, graft it into a bovine bovine antibody, which, as you may know, have these really long heavy chain CDR3s in a nutshell. I'll expand on that later when I talk about our bovine-based library. And they took that grafting antibody and showed that it bound to CXCR4 transfected cells, as well as retained the inhibitor antagonist function of the original peptide, but now in an antibody form. So this really showed this concept that you could take a peptide and graft it in heavy chain CDR3 and then create an antibody that had antagonist functions against the G protein coupled receptors. So what we wanted to do is not only take one peptide, but actually create an oligo pool of over 100,000 different motifs and graft it into the top of an antibody scaffold. But rather than use a bovine antibody, we wanted to use a fully human antibody with a long heavy chain CDR3 stem that had a huge array of different GPCR binding motifs. And that's exactly what we built in the GPCR 2.0 library. So where do those motifs come from? They came from, again, pieces or snippets of different chemokines, which are known to bind to GPCRs. Of course, GPCRs bind a whole array of different helical peptides and loop peptides. We also, as well, it's known that GPCRs actually fold over and bind themselves. So their internal domains oftentimes interact with their second extracellular loops. So we took a lot of motif content from the internal domain. And then finally, it's known that there's also, even though they're difficult to find, a fair number of GPCR binding antibodies out there. And so we took a lot of information and motifs derived from those antibodies as well and incorporated it into the final library. So after about a year's worth of work, we looked at a number of different scaffolds. Again, these are all human-derived to basically hold this diversity. We did a huge amount of work to look at displayability of different heavy chain light chain pairs, as well as long heavy chain CDR3 stems that could accept these motifs. And then once we felt confident that we could display a long heavy chain CDR3 loop in specific human scaffolds, we then ligated in this huge oligo pool that encoded for all these motifs I just talked about into the final library. So it's a fully human antibody library with multiple heavy and multiple light chain scaffolds. It has a very large motif library in heavy chain CDR3, and then we transformed it, again, in an scFv format to give us a library of over 1 billion different antibodies that have different motifs in their heavy chain CDR3 loop. So just as an example of some data that's generated from this library, one of our very first targets, which is actually the subject of a paper that's going to be out pretty soon that we're going to be publishing in NABS, is when we panned the library against GLP-1 receptor. So when we did that, we were actually able to find a number of antibodies that bound with high affinity to GLP-1R overexpressed cells, and we did the panning again on cells that overexpressed GLP-1R. Using that approach, we came up with a whole battery of different antibodies that bound with high affinity to that target, and I'm showing you the data derived from one of those on this specific slide. So shown on the left is a cyclic EMP inhibition assay, where we show that at high concentrations, our antibody actually inhibits the activation of GLP-1, GLP-1R signaling. So you can see if you go look at the black line, where that's just the activation of the receptor using the known ligand, and then in the presence of a high concentration of the antibody, you can see a nice shifting of the curve to the right, indicating we have some inhibition of signaling. If we do a full titration of the antibody up to high concentrations, you can see that we can completely inhibit the activation of the receptor with an IC50 in the nanomolar range. And then finally, we did several in vivo studies. Shown here is just one of them. GLP-1R is actually involved in glucose maintenance, and if you inhibit it, you can actually maintain high level of sugar in animals that are pre-dosed with the antibody. And so shown here in this glucose tolerance study that we did in response to antibody therapy, we were able to show that our antibody actually, again, as expected, was able to maintain a high level of glucose, shown in purple in this in vivo study. We've also shown that this antibody has a very long half-life, with a half-life of over a week in rats, indicating that even though it's an unusual antibody and that it has a motif that binds to GLP-1R, we're able to show that these antibodies have very similar half-life to any other antibody you may be using in your own particular discovery studies. The second library I'll talk about is our GPCR 3.0 library. One feedback we got about our GPCR 2.0 library is that some folks were a little thought adding in motifs might be a little bit too risky for them, so we wanted to focus in on to create a library that just used antibody-based diversity rather than motifs that bind GPCRs. So we mined the patent database and actually pulled over 61 different antibodies that bind 22 different targets, and they span a number of different germline heavy and light chain frameworks, and we've actually picked two from all those sequences, and so basically created a library that starts with that design set with those particular germline scaffolds to create a fully human antibody library derived from those sequences. And shown here is just one example of an antibody we pulled out in that panning we did against CXCR5. We show that this antibody, which again is fully human, doesn't have any motifs, it's derived, this ligand, the library is derived from that initial design set. We show that this lead has high affinity binding to CXCR5 on CXCR5 transfected cells. And we also show that the orange line on the right, TB83-107, actually also inhibits the signaling of CXCR5 in response to CXCL13, which is its known ligand. You can see that we're able to shift the activation curve to the right. The next library I'll talk about is our Hypermune library. It's a fully human antibody library. Again, it's a Fab library. And again, here I wanted to do something that no one else had ever done before, which is to basically create an oligo pool that encoded for a huge amount of diversity in heavy chain CR3. If we believe the dogma that heavy chain CR3 is the primary determinant in binding, we can. That's why we really focused in on creating and putting as much diversity as we possibly could into this one region of the antibody. So that's exactly what we did. We created an oligo pool of over 2.5 million sequences. We, of course, put diversity into the other 5 loops of the antibody as well, and we put that all into a single human germline scaffold, VH3-23, VKAPA139, and we created this fully human Fab library shown here with a diversity of over 10 to the 10th. Shown here is just one example where we used this against SARS-CoV-2, and we were able to find a number of antibodies that had nanomolar to even picomolar binding to either S1 or S1 trimer, really showing the power of the platform to pull out very high affinity as well as specific antibodies to the SARS-CoV-2 spike protein. And these antibodies also showed great inhibition in terms of blocking the S1 ACE2 interaction. So I know this is a pretty complicated slide, but basically it's a cell-based assay where we're able to show in the middle panel that these two antibodies, TB1823 and 4, show nice blockade of S1 ACE2 interaction. And down at the bottom, we actually looked at all of our antibodies that came out of this campaign from a number of our other libraries as well, and we're able to show that we had a number of antibodies that completely block the interaction between S1 and ACE2. And then moving on, we also have, as I mentioned, a whole array of different VHH libraries. I won't go into them in depth today in the interest of time, but basically we have one library that's called our VHH ratio library. We started out from these libraries from the same design set, which is a very large llama alpaca database where all of the members of the database have known antigen specificity. So all the VHHs bind a specific target, and we use that as a design set to build the 4 libraries I'll talk about today. The first one is our VHH ratio library. It takes these 3,000 VHH sequences, and we basically created oligo pools that constitute the ratios of amino acids that are allowed within that design set. And then we put them all into a consensus llama scaffold in the library. The second library is what we call our VHH shuffle library. So rather than use ratios of amino acids in those oligo pools that encode the library, like in the ratio library, we actually just use the explicit CDRs themselves derived directly from the database and just shuffled them in a unique context in this VHH shuffle library. And again, we put that into a consensus llama framework. And then finally, moving on, our third VHH library is our VHH human shuffle library. It's in a fully human scaffold, unlike the previous two, which were in a consensus llama scaffold. And again, we did the same type of shuffling approach where we just shuffled the CDRs directly from the database, but now in this humanized VHH scaffold. And then finally, using the diversity from the Hypermune library that I talked about before, we swapped out the CDR3 diversity, which previously for the VHH human shuffle library was llama derived. We switched that out with human heavy chain CDR3 diversity. So this library not only has humanized scaffold, but also has human heavy chain CR3s and the CR3 register of the library. And we've been able to show this, that these VHH libraries give us very potent high affinity antibodies to a number of different targets. Shown here is just some example work that we've done against TIGIT, where we panned the first 3 libraries against this specific target, came up with over 50 unique VHHs that bound TIGIT from each library. Shown on the bottom is just the CR3 length diversity for these 3 libraries. You can see they're very different from one another, indicating that we have very different sequences coming out of each library. And then on the right, we've actually, again, determined affinities of all these VHHs from these different targets. And so shown is the kind of different affinity bins, binders that we see coming out of this panning. And so we basically saw that we're getting typically double-digit nanomolar for this target, maybe even triple-digit nanomolar KD values. And we have over 51 variants that have affinities of greater than 100 nanomolar KD. We also did use these same libraries against the SARS-CoV-2 S1 antigen. And in this case, in the case of this target, we're actually able to find antibodies that had single-digit nanomolar, maybe even picomolar binding to the SARS-CoV-2 S1 protein. Shown here is just an array of those different antibodies that came out of the library, our Cartera LSA platform, and you can see that we have some examples of some low affinity antibodies all the way to even some picomolar binders up in the top left corner of the array. And then when we took these VHHs that bind the SARS-CoV-2 S1 protein into that competition assay I showed before, we were able to find a number of antibodies that had sub-nanomolar IC50 values in this competition assay. And we've actually taken these antibodies even further forward and shown that they neutralize live virus very well as inhibit and neutralize the virus in vivo as well. The next library I'll focus on is our structural antibody library, as I talked about before. It's derived from antibodies that are in the PDB. And again, we took antibodies from this database to basically enable us to focus in and create a library on that diversity of antibodies that have been crystallized, again, with this idea that antibodies that have been crystallized are very stable and potentially very debuggable. So shown here is the overall design of that library. So again, it's a VH3-23 VKappa139 fully human synthetic library. These are the number of oligos in each of the different regions of the CDR loops. And you can see we're using roughly hundreds of antibodies in each different loop and then shuffling them in unique contexts in this scFv library. And this allows us to, again, at the end of the day, have a theoretical diversity of over 10 to the 13th, and our final capture diversity from this library is on the order of 10 to the 10th. And shown here is some examples of how when we panned and screened against CD3, we're able to find a number of antibodies that had human cyno cross-reactivity, again, showing the power of this library. As you may know, finding antibodies that have human cyno cross-reactivity is actually a very difficult thing to do. So now I'll spend the remainder of my time introducing our 3 new scFv libraries in the library of libraries. I've actually talked about these before in a single slide. It's kind of upcoming attractions. But now we actually have some data associated with them, as well as some new names that I'm going to roll out today. The first one is what we'll call our Ancestral library. It's a library based on antibodies and the public patent database. And as you'll see later, it's a really robust library where we basically shuffle CDRs derived from patented antibodies in different contexts, again, to give us antibodies that have unique specificities to different targets. The second library is our AI Hypermutated library. It's an AI augmented library designed from knowledge of DNA rearrangement and somatic hypermutation, where we actually, in collaboration with Deep CDR, created this huge array of simulated sequences, much like your natural human immune system would do. And then we use those sequences to design, as a design set, to build a very large fully human synthetic library. And then lastly, in collaboration with a company called Taurus, we created our Minotaur library, which is a fully human antibody library that incorporates long bovine heavy chain CDR3s. And as you'll see today, it also is yielding some nice hits against different targets. Okay, so now for that first library called our Ancestral library. Again, this was actually a library that we did in collaboration with a company called Natural Antibody. And again, here we're using VH3-23, VK139 fully human germline scaffolds, and then we basically create oligo pools that constitute diversity in all 6 different CDR loops within the antibody. You'll see in a bit how we're able to come up with those diversities derived from that initial input data, which is again the patent database, but shown here is the number of oligos that went into each of the different loops. Of course, we removed any sequences that had manufacturing liabilities. The final theoretical diversity of the library was on the order of 10 to the 13th, and our final generated library was on the order of 1 billion. So how did we come up with those sequences that are in the library itself? So again, our input were VH3-23, VK139 antibodies from the patent databases, which again, we choose those scaffolds because they're many of the approved antibodies in the market today actually have those fully human germlines. We take those sequences, remove all the liabilities, and then we did a whole series of bioinformatic analyses to look at those sequences to understand how similar they were to one another. And we reduced the complexity of those sequences by looking at clusters of sequences based on Levestein distances from one another, and was able to reduce the number of CDR's sequences to a lower dimension of complexity. And we basically chose representatives within each of those different clusters to then put into the final library. So at the end of the day, shown on the right, in step 5 is the number of clusters or representatives of those sequences that were used in the final library. And if we choose to, we can actually always go back to that original data set and find other representatives to potentially optimize these leads further because we have that huge amount of information in each of those different clusters to build upon. Shown here again, we used SARS-CoV-2 S1 as a nice proof of concept antigen. We show that we can have hundreds of antibodies that bind this specific target. We also show that antibodies derived from this library are always also similar to the VHH as well as hyperimmune hits, show really potent inhibition of the SARS-CoV-2 S1 spike ACE2 interaction. And then finally on the far right, we're just showing some KD values again derived from our CARTERA platform, and you can see that we're seeing again sub-nanomolar binding to the SARS-CoV-2 S1 protein. Sorry, nanomolar binding, not sub-nanomolar. Secondly, our second new library, which is our AI Hypermutated scFv library. Here, unlike the previous, we're actually using multiple human heavy chain as well as light chain scaffolds. Here we also did something quite unique, where we actually, again, we started with this database of over 100,000 simulated antibody sequences. And here, unlike in our previous libraries where we naturally shuffle all the 6 CDRs at once, here we're actually able to take advantage of our long oligosynthesis technology, and we're actually able to make oligos that encode for heavy chain CDRs 1 and 2 as well as light chain CDRs 1 and 2 together. And so this allowed us to reduce the overall theoretical complexity and put a lot more diversity in heavy chain CDR3 by synthesizing heavy chain CDR1 as well as light chain CDR1 and 2 together in individual oligo pools. And so by doing so, we can actually build an scFv library that actually only has 4 oligo pools rather than 6. So here we actually then use those oligo pools to build the 4 separate sub-libraries shown here, where we have either VH3-23 or VH1-69 paired with either VK1-39 or VK3-20. And so we pair those into these 4 different sub-libraries. Again, the theoretical diversity of all these libraries together is on the order of 10 to the 13th. And then the final generated library that we're currently using, of course, we'll probably try to see if we can increase this further, is on the order of about 10 to the 9th. And again, the cool thing about this library is it starts from these simulated sequences derived from this neural network. And we're also taking advantage of some new library technology where we can actually make long oligos that encode for CDRs 1 and 2 for both the heavy and the light and keep the linkages between CDRs 1 and 2 together. This just shows you some of the computationally derived sequences that were used as that initial design set, again in collaboration with CDR. And so they gave us sequences that they simulated derived from naturally occurring databases of human antibody sequences. And as you can see, the length diversity as well as amino acid diversity of the sequences match up really nicely with naturally occurring sequences from human antibody databases. And then when we look at the performance of this antibody library, again, similar to the Ancestral library I showed before against SARS-CoV-2 S1, we see a huge array of different antibodies that bind in ELISA. When we make those antibodies, we see that many of them are nanomolar, even double-digit nanomolar binders to SARS-CoV-2 S1, and they also inhibit very well in that competition assay as well. Again, indicating that we have a really nice antibody library in our hands that we can again use against any target, but it's really nice validation data from the SARS-CoV-2 S1 proof of concept study with this library. Then finally, I'll end with the Minotaur library. Again, it starts with taking advantage of diversity derived from cow antibodies. And as you may know, I'm showing here on this upper top right, cow antibodies have these very long heavy chain CDR3 sequences, and they actually bin into kind of two different lengths. One is kind of long sequences that have on an average of about 25 to 30 amino acids in length. And then there's also examples of ultra long heavy chain CDR3s, which have lengths upwards of even 16 amino acids. What we wanted to do was create a fully human antibody library that focused in on the long sequences, not the ultra long ones, and then put them into different sub-libraries that either had cysteines or didn't have cysteines that are shown here on these three different sub-libraries here. So in the first one, which is library 1A, we basically pulled all the long heavy chain CDR3s that didn't have any cysteines in their heavy chain CDR3s and created this library shown here with a huge amount of diversity in heavy chain CDR3 that is devoid of any disulfides. The second one is built where we pulled all the heavy chain CDR3s that have a disulfide built into the heavy chain CDR3. So again, long heavy chain CDR3s with a disulfide was our second library, which we call the even library. And then finally, it is known that several bovine antibodies actually have an odd number of cysteines in their heavy chain CDR3, and then they pair that with another cysteine in the framework or even in CDR2, where they can form that disulfide. So we call that our odd library, and where you can actually form another disulfide with the framework itself. Similar to our previous libraries, we removed all liabilities, including unpaired glycosylation sites, deamidation hydrolysis sites. And then the theoretical diversity for all these libraries is shown in the bottom right-hand corner with an overall theoretical diversity of over 10 to the 20th. And then the final library sizes are also indicated in the bottom right. This just shows a little bit of data that we've done against panning against human CD3. As I mentioned, finding human cross-reactive clone against CD3 is a very difficult thing to do. And again, we were just really happy to see even just binders coming out of this library because it's a very unique concept of using bovine CDR3s in the context of a human scaffold. Here again, you can see we got hits from our odd library, which again is forming a disulfide with the framework. And we also see binders to our even and zero libraries, cysteine libraries as well. And since I have a little time, I'll briefly go over our TAO platform. So again, for our optimization platform, we can basically take an input antibody derived from any source, whether it's a murine antibody into a fully human antibody. We can put that into our custom software, which has a very large human NGS database. Antibody database is part of it. And what the database does is it can look at your sequence and basically pull in any fully human heavy chains, fully human CDR sequences that are similar with a defined Levenshtein distance of your specific CDR sequences. We can then synthesize those CDRs and create a custom focused library where we shuffle that natural human diversity in your fully human antibody scaffold. If your antibody is a mouse antibody, we can also humanize and create a humanization library at the same time where we're shuffling natural human diversity in the context of a humanized scaffold. And shown here is just a little bit of data where we went through that process with a low affinity yet functional PD-1 antibody where we put it into the software, created a whole series of oligos to build a library. We then put that library into phage display, candidate and screen against that specific PD-1 target, and found and was able to optimize and increase the overall affinity of the antibody over 72-fold to give us an optimized antibody of 4.5 nanomolar. And that antibody actually rivaled the PD-1 antibodies that are on the market today. So just to conclude, ways you can work with Twist Biopharma. Of course, we're very open to licensing the entire library of libraries, and that's actually our preferred way to license it. We license them on a subscription model, so that allows you to access all of the libraries. But if you choose to, you also can license individual libraries as well, although it's much more economical just to take the full subscription. Second is, of course, we have a full team of really talented antibody engineers that can basically partner with you using the libraries and pan and screen one or even multiple libraries derived from libraries against any target you wish. Again, getting back to our differentiation I talked about before, this amazing ability to build libraries so quickly and so have so many of them at our disposal really gives us a great differentiation against our competitors. And so oftentimes when a company comes to us and gives us a new target, we oftentimes will use multiple libraries and want some pools, oftentimes pooling them based on scaffold to give us a huge breadth of diversity going into our discovery projects. The third way you can work with us is, of course, in antibody optimization using the Tau platform I just talked about. So you can give us a lead derived from any source, and you can create a custom library that allows you to optimize your lead further. We're doing a huge amount of work as well to validate our libraries, as I just showed in this presentation. That is generating some leads that might be of interest for others to look at, and so we are also considering licensing out specific leads since we're not a full-fledged R&D discovery and development organization quite yet. So if any of the leads that I talked about briefly today are of interest to you, please follow up. This way is that we do, of course, a lot of people come to Twist to build their own custom libraries separate from the work that we do in Twist Biopharma. But oftentimes, if they're smaller companies, don't have the ability to screen them, maybe they don't have the expertise in phage display like we do. We can actually partner with them. They can put their order into the custom library team separately from us, and then we can take that library from the library team and actually screen it for them, oftentimes in phage display. So this allows smaller virtual companies to access the great expertise and lab facilities at Twist. And then finally, if you're interested in making a lot of antibodies, if you're actively working, again, it doesn't need to be phage display, it could be any other platform, even single B-cell cloning. If you have this need to make antibody genes as well as make antibody protein as part of your workflow, we can definitely help you out there. So if you have any high-throughput IgGs, please follow up. And that's the end of my talk. Thank you for your time and attention, and I'd be happy to answer any questions.
Great. Thank you, Aaron, for an excellent presentation. I see that we've got a few questions that have come in already, but we'll give the rest of the audience a moment to enter any other questions in the Q&A box to the left of the slides. Before we go to those questions, I'd like to thank Twist Bioscience, Gyros Protein Technologies, Specifica, Applied Biomass, Rapid Novor, Berkeley Lights, Intertech, and Ajinomoto for sponsoring this Digital Week. Also, be sure to check out the resources list to the right of your screen, where you can download a few featured white papers. All right, now, Aaron, let's go back to the Q&A, and I will go ahead and read off some of the questions that have come in, and we'll go from there. So one question I see in the pod, have you tried out your libraries using any other form of display technology aside from phage display?
Great question. So we're definitely trying to see if we can move into mammalian display. So we're working actively to create a CHO-based platform in the very near future. And you may have seen that we announced a couple months ago that we did a licensing deal with Applied Stem Cell. They actually have a CHO-based platform with a single landing site that you can create a library into. And so we'll be creating a mammalian display platform very soon. So be on the lookout for that, and hopefully at the end of the year we'll be able to show some data associated with that work. We Next question I see, do you have any toxicity data for the antibodies you selected? We do have a little bit. So I didn't get into it today, but as I did mention it briefly, that we have a number of antibodies that show activity and neutralization against the SARS-CoV-2 virus. We've actually gone in vivo with a number of those antibodies, and as part of that, done kind of a limited assessment of toxicity. And we, again, don't see any toxicity against in those models. Of course, that's against an infectious disease target where we're really targeting the virus rather than the normal biology of the animal that we're going into, but suffice to say we don't see any toxicity associated with those antibodies, as well as in the GLP-1R case I showed before. In that case, we actually are hitting the naturally target in the animal, and it does have mouse cross-reactivity. And again, in those cases, we don't see any appreciable toxicity associated with dosing those animals in a multi-dose fashion.
Okay, perfect. Next question. For the VHH human library construction, do you have a step where you check for aggregation?
It's hard to kind of check the state of the library for aggregation as we build it. We, of course, can remove sequences that might lead to developability risk, which is what we do for all of our libraries. So that's one kind of check we do on upfront as we build it. But again, then we're shuffling the diversities in those CDR loops together in different contexts. And then on the back end, after we come up with initial hits, we do put those antibodies through a battery of tests for aggregation, stability, non-specific binding. So we do a little bit of work on the front end, but we also do a huge amount of work on the back end to make sure that the leads coming out of the library are very developable.
Great. Another question. What are the CDR3 lengths in the Hyperimmune human VHH library?
Yeah, great question. I kind of jumped over it a little bit. So again, the CDR3s in the Hyperimmune VHH library are derived directly from heavy chain CDR3 diversity. It's the same heavy chain CDR3 diversity as in the Hyperimmune library Fab library I talked about before. And so the average lengths are what you see in nature. So it's around 12 to 13 amino acids average length.
Great. Another one. Why did you only humanize part of the frameworks in the VHH in shuttle?
Great question. So again, we can only partially humanize the VHH framework because we need to retain some of the mutations in framework 2 that lead to the solubility of the VHH itself. If we actually did a full humanization of the VHH domain, we might have some issues with aggregation of the VHHs, because as you probably know, heavy chains almost invariably have this kind of hydrophilic patch where they pair with the light chain. So we need to keep some of those mutations and those sequences derived from the llama framework to ensure that the VHHs stay soluble and don't aggregate.
Perfect. Another one, do you use trinucleotide mixes to create the diversity in the CDRs? And if not, how do you actually make sure you get no frame shifts in the library?
Yeah, great question. So again, everything we do starts with this silicon platform where we basically make individual oligos at discrete length and sequence. So we don't actually have to use trinucleotides. We can just use natural, just regular nucleotide synthesis to create the oligos. And then we basically can create a pool from even 10 oligos, even up to 1 million oligos of discrete sequences. And since we're synthesizing each sequence separately, that gives us a really high level of quality of the oligos that constitute those pools. And so as a result, we don't really often see a lot of errors or even frame shifts of sequences in those oligo pools. When we then take those oligo pools and put them into a library, we also then do NGS sequencing on the back end of that library build and ensure that all the oligo pools are coming together in the right fashion. And so that allows us to really ensure that the quality of the library is much higher than what you would ever see from a library derived from using trim-based oligos, because we don't have to worry about trinucleotides coming together in a specific manner. Here we're just making sequences using natural oligonucleotide synthesis technology, and that gives us a really high level of fidelity of the DNA in our antibody libraries.
Perfect. So another question, would it be possible for you to offer troubleshooting help for university labs who are trying to build their own synthetic human VHH libraries?
Yeah, the library team that we have at Twist is top notch, and they've done a huge amount of work with others that have built VHH libraries in the past, and so they can definitely help you out as you build your library. I think you can just go to them with some ideas, maybe some design sets you might already have in mind, maybe even have some literature or papers you might want to pull from, and they can really help you in that overall design process. And you have a great project management team as well that can kind of guide you through that process.
Perfect. How do you come up with new libraries in the library of libraries?
Yeah, they come up from a number of sources. Of course, we have a lot of ideas internally within the antibody engineering team. But as you've seen today with the 3 new libraries I talked about, we also do a lot of collaborations with top-notch companies that have a lot of expertise in antibody discovery. So for example, the 3 libraries I talked about today were derived from collaborations with Natural Antibody Library, Deep CDR, and Taurus. And so we were very open to working with others to create libraries together. So if there's anybody out there that wants to build an antibody library together in collaboration with Twist Biopharma, please see us, and I'd be happy to work on our library together, and where we can actually both go out and use that library collaboratively.
Great. Just for attendees still listening, I see there's 2 more questions in the pod. We got a few more minutes, so if you have any questions, feel free to go ahead and keep adding. Aaron, here's the next question. How do you see the HT-IgG offering expanding in the future?
Great question. So some ways to potentially expand the high-throughput IgG offering would be to look at other scales. We are also thinking about potentially moving from not only doing HEC-293, but also thinking about doing CHO-based antibody production, since oftentimes your final lead is oftentimes made in a CHO-stable cell line and not in HEC-293. And then we also want to see if we can, decrease the turnaround time as well in our process, and we're trying to think about ways to make DNAs even faster through our process, because again, a big chunk of that time in the high throughput IgG process is actually making the DNA itself. So, and again, there's a lot of innovations on trying to decrease turnaround time at Twist, and that's going to have a huge impact throughout the company as well.
Great. And the final question I see is, how do you take full advantage of your library of libraries in antibody discovery projects?
Great question. So as I mentioned before, when we do a new project for our partners, we oftentimes we take full advantage of libraries by just pooling libraries into different scaffold mixes. So we can basically take the Fab library, all the single chain libraries, and then the VHH libraries, and we can pull them together. And so that really allows us to take full advantage of libraries, because we can basically have these huge pools of individual libraries that we're pulling together, and gives us, even tens of billions of different antibodies in each of those different pools. So that allows us to really ensure that we give the results back to our partners, that we have antibodies derived from multiple libraries that potentially bind to different epitopes and are derived from different sources. So that gives us a huge advantage in using the libraries for our discovery projects. Perfect.
Aaron, it looks like that's the last question in our chat pod, so I want to thank you today for a great session. For anyone still listening, I want to just remind you that the session was recorded today. You will receive a notification within 24 hours when the on-demand session is available for viewing, if you wish to view it again or listen to it again. Before you log off, we ask you to please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. And Aaron, thanks again for the talk, and everyone else on behalf of Informa Connect Life Sciences. Hope you have a great day. Thank you.
Thank you, Michael. Thanks, everybody.